Greetings all, I'm Farizul Fahmida from the Department of Microbiology. I'm here to present about the topic Preservation of Industrially Important Microbial Strain. Objectives of microbial preservations are to prevent the genetic drift, mutation accumulation or plasmid loss that could compromise the strain's industrial performance, to ensure high recovery rates of metabolically active cells when needed for production, to maintain pure culture free from contaminating organism, to preserve the specific metabolic capabilities such as enzyme production, metabolic yield, and substrate utilization, to meet good manufacturing practice requirements and maintain strain traceability. The primary goal of preserving industrially important microorganisms is to maintain their unique characteristics for future use while preventing the genetic and physiological changes. Methods for microbial preservations include two methods. They are short-term preservation method and long-term preservation method. Short-term preservation method is an active metabolic state which consists of three methods. They are periodic subculturing, refrigeration, and mineral oil overlay. Whereas long-term preservation method is a suspended metabolic state and consists of two methods, lyophilization and cryopreservation. Cryopreservation at minus 80 degrees Celsius or at liquid nitrogen. The first one is short-term preservation method, which is an active metabolic state. These methods maintain microorganisms in a metabolically active state and are suitable for frequent access over weeks to months. The first is periodic subculturing. The principle of periodic subculturing is to regularly transfer the fresh media before nutrients are depleted. Then transfer cells to the fresh agar slants or broth every 2 to 8 weeks depending on the species. It uses minimal growth condition to reduce the metabolic activity and prolong intervals between transfer. It has a high risk of genetic drift, contamination and phenotypic changes labor intensive. Second one is refrigeration. The principle of refrigeration is to store the slants at 2 to 8 degrees Celsius, which slow down the metabolic rate without stopping it completely. Incubate until good growth appears, then seal with parafilm and refrigerate. The duration of refrigeration is 4 to 12 weeks for bacteria and 4 to 26 weeks for fungi. And the application is which is short-term maintenance of frequently used strain. And the limitations include media dehydration, gradual loss of viability, and not suitable for long-term genetic stability. The third one is mineral oil overlay. The principle of mineral oil overlay is a layer of sterile mineral oil creates oxygen-limited conditions by reducing the metabolic activity. The procedure for mineral oil overlay is growth culture on agar slant until late log phase and add sterile mineral oil to cover the slant completely, which is 1 to 2 cm above the agar surface, and then store upright at 15 to 20 degrees Celsius. The duration of mineral oil overlay is 1 to 5 years for many bacteria and fungi. It is very simple and low cost, which extends the subculture intervals significantly. The disadvantages of mineral oil overlay is it is messy retrieval and it has a potential for contamination during sampling. The second method is long-term preservation method, which is a suspended metabolic activity. These methods halt or dramatically reduce the metabolic activity, enabling the storage for years to decades with minimal genetic change. The first one is lyophilization, which is also called as freeze drying. The principle behind the lyophilization is removal of water from the frozen culture via sublimation under vacuum, and their components include skim milk 10 to 20%, sucrose 12%, trehalose or glutamate, which acts as protective agents. And it has a rapid freezing at minus 40 degrees Celsius to minus 80 degrees Celsius. Primary drying involves sublimation under vacuum, and secondary drying involves removal of water bound at slightly higher temperature. They are sealed under vacuum in ampoules and stored at 2 to 8 degrees Celsius or at room temperature. They are viable for about 10 to 50 years for many bacteria and yeast. Their success rate is 60 to 95% depending on the species and protocol optimization. The second one is cryopreservation. The principle behind the cryopreservation is storage at ultra low temperature where metabolic activity ceases. There are three methods. First is standard freezing, which has a temperature range of minus 20 degrees Celsius to minus 40 degrees Celsius. And the duration of standard freezing is 6 months to 2 years, and it is used for medium term storage of robust organisms. And the second is ultra low freezing. It has a temperature range of minus 80 degrees Celsius to minus 135 degrees Celsius, and its duration is 1 to 10 years, and it is applied in long-term storages of most bacteria, yeast and fungi. The third is liquid nitrogen, which has a temperature range of minus 150 degrees Celsius to minus 196 degrees Celsius, and its duration is indefinite theoretically, and it's applied in master stocks of all microbial types including delicate strains. And the cryoprotectants used are glycerol, DMSO or sucrose, which is used to prevent the ice crystal damage. Controlled red freezing often yields better recovery than abrupt freezing. Rapid thawing in 37 degrees Celsius water bath, which is used for maximum recovery. And the advantages include highest viability recovery for diverse microorganisms and it has excellent genetic stability. And the disadvantages include continuous energy requirement and potential for storage system failures. The overall methods for preservation of microbial strain involve two methods. They are short-term method and long-term method. Under short-term method, there is a refrigeration method, which comes under the principle that slow down the metabolism at 2 to 8 degrees Celsius, and its typical duration is about 1 to 6 months, and it is best for working strains that is used for daily. And the major considerations are it is simple but risky for long-term stability. And the next one is mineral oil overlay, which forms under the principle and it limits oxygen on agar lands. It has a typical duration of 1 to 5 years. It is best for extending subculture intervals. And the major consideration are it has a high risk of contamination on sampling. Next one is long-term method. The long-term method, the first method is lyophilization, which comes under the principle by removing water from frozen culture, and it has a typical duration of 10 to 30 plus years. It is best for master stocks and easy distribution. The major considerations are it has high setup cost and not all microbes survive. The next one is cryopreservation at minus 80 degrees Celsius. The principle is it halts metabolism with cryoprotectants. The typical duration is about 1 to 10 years. It is best for most bacteria, yeast and fungi. The major considerations are it requires reliable minus 80 degrees Celsius freezer. And the next one is cryopreservation by using liquid nitrogen. The principle is it stores at minus 196 degrees Celsius. The typical duration of cryopreservation is it is virtually indefinite, and it is best for all cell types and it has ultimate security. The major considerations are it has highest cost and requires liquid nitrogen supply. Special considerations for microbial storage are bacteria and yeast are generally robust and most methods work well, whereas filamentous and actinomycetes are often sensitive to lyophilization, and so cryopreservation with specific cryoprotectants like 10% sucrose is preferred. Anaerobic microbes require oxygen-free handling during preservation and revival. Recombinant strains uses cryopreservation in liquid nitrogen. It is best to guarantee the genetic stability and prevents the plasmid loss. use cells from the late logarithmic growth phase, use cryoprotectants such as glycerol or DMSO, which is essential for frozen stocks, and avoid repeated thawing and store in small single-use aliquots. Always check the viability and key industrial traits after preservation. The emerging technologies which are primarily used to enhance the strain's genetic stability, productivity, and resilience before long-term preservations are CRISPR or Cas9 gene editing. This technology allows for precise, targeted modification to the microbial genome. Example, gene knockout or insertion, which is to improve the specific traits such as increased yield of a target product or enhanced stress resistance. And the second one is metabolic engineering. This systemic approach involves modifying specific biochemical pathways to redirect the cellular metabolism towards the overproduction of desired compounds. Examples, biofuels, pharmaceuticals, or enzymes. The third technology is directed evolution or adaptive laboratory evolution. This technique mimics natural selection in a controlled laboratory environment by applying specific selective pressures over many generations to obtain strains with desired and improved characteristics. Examples, thermotolerance and substrate utilization. The fourth technology is omic technology that involves genomics, transcriptomics, proteomics, and metabolomics. The integration of these high throughput data analysis method provides a deep understanding of complex cellular interactions, allowing for more rational and efficient strain design and optimization. Fifth technology is genome shuffling. This method uses recursive protoplast fusion to rapidly combine the beneficial mutations from multiple parent strains into a single highly optimized hybrid strain. The next is emerging physical preservation methods. First method is cold plasma method. This is a non-thermal decontamination technique uses ionized gas to inactivate the microorganisms through oxidative damage, showing promise for decontaminating surfaces without compromising the material quality. And the second technology is pulsed light technology. This method uses short, high-intensity pulses of broad-spectrum light to damage the microbial DNA and disrupts the cell membrane, offering a rapid, chemical-free sanitation option for surfaces. Third technology is high-pressure processing or pulse electric fields. These non-thermal processing methods can inactivate spoilage microbes and enzymes while preserving the quality of product. Often used in hurdle technology approaches combining multiple preservation methods. Fourth one is antimicrobial edible films and coating. Natural antimicrobial agents such as essential oil or chitosan are incorporated into the biopolymer films and applied directly to products to regulate the gas exchange, reduce the moisture loss, and inhibit microbial growth during the storage. Fifth one is nanotechnology in preservation. This involves the use of nanomaterials such as silver or silica nanoparticles as a potent antimicrobial agent in food preservation, although safety concerns require future work. And the next is emerging AI-driven preservation methods. First one is real-time monitoring, that is biosensors or IoT. AI analyzes the live data on pH, temperature and growth to detect the anomalies and predict outcomes. Second is digital twins. AI simulates the process changes in a virtual mode first, optimizing condition and minimize the real-world risk. And third is omics data analysis. AI integrates genomics, proteomics to identify the key genes, predict metabolism, and guide strain design. Fourth one is predictive analytics. ML models forecast shelf life, contamination, or equipment failure, enabling the preventive maintenance. Fifth is automated control system. AI dynamically adjusts the bioreactor parameters to maintain the optimal conditions. Sixth is intelligent strain design. Deep learning aids synthetic biology, optimizing tools like CRISPR to develop the robust high performance strains. Thank you.